Welcome to another lecture by MedicoMedics, Physiology Chapter 6, Synaptic Transmission and Neurotransmitters. So in this lecture we will talk about synaptic transmission. We will talk about its steps, mention neurotransmitters, show some examples of synaptic transmission, disorders of it, and end with a summary. Now, what is synaptic transmission? It is the process by which one neuron communicates with another or with a target cell. Now, this happens at the synapse. So the synapse is a junction between two neurons or between a neuron and a target cell, as illustrated here. Now, there are different types of synapses. We have electrical synapses where there is a direct electrical current flow, but this is rare in humans. Then we have the common one, which is a chemical synapse. This is communication via neurotransmitters, and this is the most common one. So synaptic transmission is like passing a note from one cell to the next. It's how signals travel in your body. Now let's explore the structure of a chemical synapse to understand how it works. Now, key components include the presynaptic neuron. This is where the signal is sent. Then we have the synaptic cleft, and this is a small gap between neurons, 20 to 40 nanometer wide. And this cleft is illustrated right here. So we have the presynaptic neuron, and then we have the synaptic cleft, and then we have the postsynaptic neuron, which receives the signal, illustrated here. Now, the synaptic vesicles are located in the presynaptic terminal. As you can see here, synaptic vesicles. And these contain these neurotransmitters, meaning the chemical messengers. So whatever is supposed to bind to these receptors here are contained within these vesicles. Now, we also have receptors. That's a crucial component. And these are found on the postsynaptic membrane. So this is the postsynaptic neuron, and here is the postsynaptic membrane. And you can see the illustration of these transmitters, these receptors on this membrane. So these bind to these neurotransmitters to initiate a response. So let's clear that out. We have these vesicles in the presynaptic neuron. They travel, get released. The neurotransmitters or chemical messengers inside this vesicle gets released. It then jumps into the synaptic cleft, where it then finds its target receptor, called the postsynaptic receptor, gets picked up, and whatever that message was will then be relayed here in the postsynaptic neuron, leading to some kind of a response. So these are key components and are indeed high yield. Now let's talk about the steps of the synaptic transmission. So first we have the arrival of the action potential. So it reaches the presynaptic terminal. We then have calcium ion influx. So voltage gated calcium channels open, and that allows for calcium to enter. Now with that, the neurotransmitters gets released. So the synaptic vesicles, if you remember, let's draw a painting here of the presynaptic neuron. It contained these vesicles, right? Which contained the neurotransmitters. Now with that influx of calcium, these synaptic vesicles fuse with this membrane here. And with that, they then release these neurotransmitters into, you guessed it, the synaptic cleft, which was in between here, and the postsynaptic neuron. Paint that here. Now, don't mind these beautiful drawings. They are just there to illustrate something. And you have these receptors on the postsynaptic neuron, right? So these neurotransmitters cross and bind with these receptors. Now, after that, we have we had 4 here and 5 here. So we have the postsynaptic response. So there will be either a depolarization, if it's an excitatory reaction or response, or hyperpolarization, if it's inhibitory. It will depend on this neurotransmitter and on the receptor. So the message, what is the message? Whatever that message is, it will then have a different response here. And once that signal has gone, whether it is to inhibit something or excite something, either depolarize or hyperpolarize, we then have termination. So the neurotransmitters left in the cleft after their response and their work are then removed via the process of reuptake or degradation or diffusion. So here you can also see an illustration of more steps involved. So we have the action potential arrival at the presynaptic neuron. We have depolarization of the membranes. We have voltage-gated calcium channel activation, calcium influx. This leads to the vesicles fusing to the membrane and getting released. The neurotransmitters are getting released. These neurotransmitters then move into the cleft, and then they reach their receptors, and then binding with the postsynaptic membrane, and then we have the response, either an inhibitory response or excitatory. And this is directly correlated to what kind of polarization that occurs. Is it a depolarization due to sodium influx, or is it hyperpolarization due to potassium efflux or chloride influx? And/or. Now, what are these neurotransmitters that we've been talking about, and how do they work? Let's dive a bit deeper into these chemical messengers or neurotransmitters. Now, neurotransmitters are defined as chemicals that transmit signals across a synapse. Now, we mentioned excitatory, inhibitory, or modulatory types. Now, the excitatory, their mechanism is often to open ion channels that allow for positive ions to flow into the cell, like sodium or calcium. And its role is mainly, so the role of excitatory neurotransmitter is mainly to increase the likelihood that neurons will fire an action potential. So just as the name suggests, they excite the postsynaptic cell by making its membrane potential more positive, so moving it closer to the threshold required to trigger a response. So if you remember from previous lectures, we had this number of minus 70 millivolts. And if we have an influx of positive ions, we will move minus 70 closer to 0, or even closer to, let's say, plus 20, where we have a threshold met for an action potential to go. So it makes sense then that if positive ions enter, we will move closer to our threshold. And then we have inhibitory ones. And the mechanism here is to often open ion channels, but it allows then for negative ions to enter the cell. So it's the opposite. And examples of negative ions include chloride, for example. Or, if you remember, we also had these channels, these leak channels for potassium to get out. So the mechanism of inhibitory is either the influx of chloride or negative ions, or the efflux of positive ions like potassium. And its role is to decrease the likelihood that a neuron will fire an action potential. So they inhibit. They calm down the postsynaptic cell by making its membrane more negative, moving it further away from the threshold. Again, minus 70, if you get more potassium that's positively charged leaving the cell, minus 70 will then go closer to minus 80, for example. And that gets us further away from our threshold. Hope that makes absolute sense now. What about modulatory? So these often act through second messenger systems or affect how other neurotransmitters are received or released. So these have the role of modifying the strength or efficiency of signals being transmitted. So modulatory neurotransmitters influence how excitatory or inhibitory signals are processed by altering receptor sensitivity or neurotransmitter release or synaptic strength. And we can mention some examples of each. Maybe you have heard of those. So excitatory ones include, for example, glutamate, and it is the most abundant neurotransmitter in the brain, and it plays a crucial role in memory, cognition, learning, etc. And that's an excitatory neurotransmitter, glutamate. Another one that acts as a excitatory neurotransmitter in certain contexts, like in muscle contraction at something called the neuromuscular junction, which we will mention soon, is acetylcholine. Now, what about inhibitory ones? We have one called GABA, which stands for gamma aminobutyric acid, and it is the main inhibitory neurotransmitter in the brain. It helps to regulate overexcitement and is also involved in for example, prevention of seizures. So GABA. We have glycine, which also acts as an inhibitory neurotransmitter in the spinal cord. Modulatory includes, for example, dopamine, serotonin, norepinephrine, etc. So let's better visualize this by putting it into a table to see the key differences between them. So for effect, excitatory increases the likelihood of firing, inhibitory decreases likelihood of firing, and modulatory ones adjust signal strength or efficiency. Mechanism of excitatory is to depolarize membrane potential. Inhibitory is to hyperpolarize membrane potential. And modulatory alter neurotransmitter systems indirectly. Immediate response, excitatory, yes. Inhibitory, yes. Modulatory, often slower, indirect effects. Examples of excitatory ones, as we mentioned, glutamate, acetylcholine, inhibitory GABA, glycine, and modulatory dopamine, serotonin, norepinephrine. Now, you may wonder whether or not, for example, dopamine, serotonin, and norepinephrine, are they not inhibitory or excitatory? Can they not have these effects? And the answer is yes. Why we primarily consider them modulatory neurotransmitters is because first they belong to the monoamine group, and they act largely through something called the G protein coupled receptors, rather than directly opening ion channels like classical, quote unquote, classical excitatory neurotransmitters like glutamate do, or inhibitory ones like GABA. So these are more specifically inhibitory with direct effects. But these modulatory ones, typically their effects are more long-lasting, more diffuse, and rather than generating these immediate excitatory or inhibitory postsynaptic responses or potentials like a GABA would or a glutamate would. So it's for this reason we more precisely put them in the modulatory category. However, can they be excitatory or inhibitory? Yes, it depends on the receptor type that they activate. So we have several types of dopamine receptors, for example. We have D1 receptors, D2 receptors, and so on and so forth. And depending on which receptor it's binding to, it could have excitatory, like when it binds to D1-like receptors. Or it could, we're talking about dopamine now, it can have inhibitory effects when it, for example, binds to D2-like receptors or D3 receptors. So, and the same is true for serotonin, for example. It can be excitatory when it binds to, for example, 5-HT2, if my memory serves me right here. But it can also be inhibitory if it's binding to 5-HT1 receptors. So it too will depend on the receptor type. And the same goes for norepinephrine. So primarily, why we primarily call these modulatory neurotransmitters is because they adjust synaptic activity, first of all, over longer timescales. But then, indeed, they can be excitatory or inhibitory depending on receptor type. And unlike, for example, glutamate or GABA, these monoamines, that dopamine, serotonin, norepinephrine, etc., they don't have a single fixed effect. So let's just briefly take a look at some examples of synaptic transmission. So we mentioned the neuromuscular junction. And the neuromuscular junction is a specialized synapse between a motor neuron, illustrated here, and a muscle fiber, illustrated here. Now, when an action potential reaches this nerve terminal, it triggers the release of acetylcholine in these vesicles to enter into the synaptic cleft here. And acetylcholine then binds to nicotinic receptors on the muscle cell membrane. And this then leads to an influx of sodium. And if you remember, sodium is positively charged. So we have a negative resting membrane potential, let's say, of -70. With the influx of sodium, this will move towards threshold, causing depolarization. And this depolarizing of the cell, of the muscle cell in this case, generating a action potential that will ultimately cause muscle contraction. Another example is dopaminergic pathways. So dopamine signals in the brain regulate movement and reward. Because dopamine, as we've mentioned, is a key neurotransmitter in the brain, but it's involved in several types of pathways. In a pathway called the nigrostriatal pathway, dopamine from the substantia nigra regulates voluntary movement by modulating activity in the basal ganglia. But in another pathway called the mesolimbic pathway, dopamine release in the nucleus accumbens plays a critical role in the brain's reward system. So it reinforces behaviors associated with motivation, addiction, pleasure, etc. Another one is pain modulation. So the perception of pain can be regulated by the release of endorphins. And these endorphins are endogenous opioids. So these chemicals act on opioid receptors in the spinal cord and brainstem to inhibit the release of pain-transmitting neurotransmitters like substance P or glutamate. So by reducing neurotransmitter release at the synapse, what endorphins do is they help decreasing or dampen pain signals, which leads to reduced pain perception and therefore increased tolerance. And this is seen, for example, in stress-induced analgesia or the effects of opioid drugs like morphine, for example. Now, in this lesson, the idea is not for you to memorize a bunch of pathways. You will have time to learn a lot of these as you move on in your studies. The idea here is to get a clear concept of synaptic transmission. It happens at different spaces or places. It will therefore have different effects. Makes sense. The process of how it happens is very similar, though. And this is what we want you to pick up here. As there are several steps and structures involved before we start talking about what specific neurotransmitter and what is the specific receptor, therefore what is the specific outcome, it's the structures involved that is important right now, like knowing that there is a presynaptic neuron. These vesicles reach its terminal. There is an influx of ions like calcium. This vesicle then fuses with the membrane, releasing the neurotransmitter, whatever that is. It then enters a cleft, or the synaptic cleft, to find its receptor. Once it binds to it, there is an influx or efflux or whatever of other ions, leading to contraction in this case, when we're talking about acetylcholine, for example. So get this concept clear. Now, to make it even more clear, we're going to see what happens when this system here is disrupted. Now then, let's talk about disorders of synaptic transmission. Now, you may recognize some of these quite well-known pathologies. Let's start with Parkinson's disease. So Parkinson's disease is a neurodegenerative disorder caused by the progressive loss of dopamine-producing neurons. And it happens in the substantia nigra, which is a part of the brain involved in movement control. So it makes sense then if we have a loss of this, we will have movement issues. So the symptoms we typically see, though, then in Parkinson's include, for example, muscle rigidity, bradykinesia, which is slow movement, postural instability, tremors, etc. And the treatment for Parkinson is something called dopamine replacement therapy, which makes sense because it's a loss of dopamine, right? Loss of dopamine producing neurons. We have decreased dopamine. We want to increase that. However, interesting fact, the treatment is not to give directly dopamine. And you may wonder why. Now, we have a dedicated lesson on Parkinson's, but just to make it more interesting for you, we give something called L-DOPA or levodopa when treating Parkinson's. And it's sort of a precursor to dopamine. Now, the reason for all of this is because dopamine, if given in its form, dopamine, cannot cross the blood-brain barrier. Now, if you haven't heard about the blood-brain barrier, it is a selective barrier that protects the brain by regulating the passage of substances from our bloodstream. So essentially, there is another layer between our normal bloodstream and whatever goes into the brain. And that extra protective layer, we call it the blood-brain barrier. And it has a fitting name because it is indeed a barrier, blocking things that should not go in. So if we have, for example, pathogens or whatever in our blood or toxins, we don't want them to enter into our brain. I guess we don't want them in our blood either, but we definitely don't want it in our brain. And inversely, we want essential nutrients like glucose and proteins, etc., to indeed go through this barrier and enter our brain. So it helps regulating this passage between our bloodstream and our brain. Now, why is this relevant to Parkinson's? Because when we give levodopa, levodopa actually does cross the blood-brain barrier. Dopamine doesn't. So in this precursor form, levodopa goes through into the brain. So problem one solved. And then we have an enzyme that converts levodopa into dopamine. And that enzyme, for those who are interested, is called dopa decarboxylase. And so the result will be an increase of our dopamine levels in the nigrostriatal pathway, which is then improving our motor symptoms. Because you remember in the previous slide, we had the mesolimbic pathway and we had the nigrostriatal pathway related to dopamine. The mesolimbic was more related to the reward system and the nigrostriatal with movement. Now, let's look at another one, depression. Now, depression is a mood disorder associated with reduced levels of serotonin, which is a neurotransmitter that regulates mood, emotions, overall well-being, essentially. And having lower serotonin levels can contribute to symptoms like persistent sadness, loss of interest in daily activities, fatigue, etc. What we essentially call depression. And what we do is we treat it with something called SSRIs, like fluoxetine, also known as Prozac. Now, you may wonder, why do we give something called selective serotonin reuptake inhibitors? Because that's what SSRI stands for. It's because it's in the name, reuptake inhibitor. If you remember from previous slides, we talked about the presynaptic neuron, and then we had the postsynaptic neuron. We had the release of these neurotransmitters called serotonin in this case. We have receptors where we want serotonin to bind for the positive outcome we want. Now, if it doesn't bind, it's degraded or it goes through reuptake. So there are transporters in our presynaptic neuron made for these serotonin that remain in the cleft to be recycled, to go through reuptake. And so the idea here is, what if we make serotonin stay in the synaptic cleft longer? Will that not increase the likelihood of this serotonin to bind to this receptor? Essentially increasing serotonin availability. Because remember, we have connected this disorder with reduced serotonin availability or levels. So can we reverse it? Now, instead of just adding a bunch of serotonin, what about blocking this reuptake transporter? So this serotonin cannot go through it. And then it remains in the cleft for a longer duration, leading to the increased likelihood of binding to this receptor on the postsynaptic membranes. So that's the idea of SSRIs. Now, in our psychiatry courses, we go into depth, and in our pharmacology lectures on antidepressants, but this is to give you a general idea of how it works. Now let's clean this up and take myasthenia gravis. Myasthenia gravis, which is an autoimmune neuromuscular disorder in which our body's immune system produces antibodies that attack acetylcholine receptors at the neuromuscular junction. So this can be directly correlated to the previous slide, where we talked about the neuromuscular junction, and we said acetylcholine enters the cleft, binds to nicotinic receptors, leading to sodium influx, leading to muscle contraction, right? And here we have a disorder where our immune system, for whatever reason, has produced antibodies that attack and destroy our acetylcholine receptors, causing muscle weakness. We're not going into detail of how this immune response is actually happening and the complement systems activated, leading to the destruction of these receptors. However, the essential conclusion is that acetylcholine will have a tougher time binding to these receptors at the neuromuscular junction, leading to not having the sodium influx, meaning no depolarization, meaning no muscle contraction, meaning muscle weakness. So that's the link. And what we do here, similarly to how we approach other problems, is that we give something called acetylcholinesterase inhibitors, like pyridoxamin. I think that's how it's pronounced. We also treat it with immune suppressants and corticosteroids, but that's related to depressing our immune system, so we decrease this attack. But the idea of giving acetylcholinesterase inhibitors is more specific to what we're talking about right now. Because similarly to us wanting to increase the availability of serotonin in the synaptic cleft by stopping or inhibiting the reuptake of serotonin, in the same way we want to increase the availability of acetylcholine in the synaptic cleft so it can bind with its receptor. And we do that by inhibiting an enzyme. The enzyme is acetylcholinesterase, which has the function of degrading, essentially breaking down acetylcholine. So we stop this enzyme from functioning, or we inhibit it, leading to what? Less degradation of acetylcholine, leading to increased availability of acetylcholine, increased likelihood of it binding despite the decreased number of receptors due to immune destruction. Hope that makes sense to you. And lastly, we have epilepsy, which is overactive excitatory signaling leading to seizures. Epilepsy is a neurological disorder characterized by recurrent seizures due to excessive and uncontrolled excitatory signaling in the brain. And this hyperactivity can result from imbalances in neurotransmitters such as glutamate, which was excitatory, and GABA, which is inhibitory. Of course, seizures can vary in type, severity. They can be very brief, and they can be full-body convulsions. So treatment would be anti-epileptic drugs like valproate to stabilize neural activity. So hopefully it's starting to make sense now by also illustrating with these different examples, all of them having different effects and outcomes. One is connected to muscle movement issues, and it's related to dopamine. We have a loss of dopamine. We have certain symptoms. Another one is our mood linked to reduction of another neurotransmitter. We approach it by inhibiting the uptake, increasing availability, increasing likelihood of firing. In the case of Parkinson's, we can't give dopamine, so we give levodopa. We know that it enters into the brain where it's converted into dopamine, helping us with this movement issue or with these motor symptoms. Another one, myasthenia gravis. We've identified that it's our immune system destroying these receptors. We understand what acetylcholine is supposed to do. And with its absence, we have muscle weakness. So we stop whatever is breaking it down, leading to increased availability, leading to better muscle contraction, etc. and etc. So, in summary then, synaptic transmission is crucial for neuron to neuron and neuron to target communication. Neurotransmitters drive excitatory, inhibitory, or modulatory effects. Disruptions in synaptic signaling are linked to neurological and psychiatric disorders. And that's the end of this lecture. Thank you for listening. Continue now to Chapter 7.